more about cancer biology. So what we talked about on Thursday was getting into why genetics is important. And again, I consider cancer to be a genetic disease, meaning it is caused by accumulation of mutations in specific tumor suppressors and oncogenes. And so therefore, because it's mutating the DNA in those cancer cells, it's a stable and inherited at the cellular level phenomenon. And the second point here that probably was made clear on Thursday was most tumor cells have numerous chromosome aberrations. They can have deletions and amplifications. We talked about I showed you examples of a lot of different ones. And point mutations even that you can't see. And so again, those are all impacting tumor suppressors and oncogenes and other proteins that are ultimately involved in cancer. So we're going to move from that background into, yeah, Troy.
Sorry, I wanted to see you describing it as stable, but you described it previously as unstable.
So the genome is unstable, meaning that it can keep acquiring more mutations, but stable in that the cancer has mutations in it that are going to be passed on from one cancer cell to the next cancer cell. So it won't, so if you can think about it in terms of evolution, once this cancer cell kind of acquires the mutations in order to proliferate and grow and not die and all those things, it's probably going to keep them and not lose those when it divides into another two daughter cells. And so those daughter cells are also going to have those mutations, and that's what I meant by, yeah.
So stability and kind of being able to stay alive.
Stability and being able to maintain those genetic abnormalities that are supporting its being a cancer cell. Yeah, exactly. Yeah, I realize that genomically unstable is maybe a little complicated. So once we get to that part, talking about that hallmark, the idea there is that normal cells have DNA repair pathways that are so, really trying to keep the genome normal. But the cancer cells often have alterations in DNA repair so that there's a little bit more fluidity, a little bit more opportunity to get mutations. Yeah. Okay, good question. So today we're going to get into the causes of cancer, really which is etiology of cancer. What kinds of things can contribute to cancer? And the outline is shown here. Etiology, again, etiology is basically causes of cancer. And we'll talk a bit about we'll talk a lot about epidemiology, what is epidemiology, and how that applies to cancer biology. The concept of environment versus heredity contributing to cancer. And then we'll spend the rest of the time, most of the time, talking about chemicals as carcinogens. And as this says here, most of these are electrophiles. Many of these carcinogens are also mutagens that mutate the DNA. That makes sense. And we'll talk about two different examples of things that are called procarcinogens, benzoylpyrene and aflatoxin. And probably then we will get to this part, talking about physical carcinogens that can act as mutagens, the Ames test to test whether something is a mutagen, and then finally some non-mutagenic hormones that can act as carcinogens. And the example will be diethylstilbestrol, which we'll get to maybe on Thursday. We might not get that far today. Okay, so epidemiology. So epidemiology can apply to cancer. In this class, we're thinking about it in terms of cancer, but this could apply to any disease. And simply stated, it's up there on the slide. It's the study of the distribution and prevalence of a disease. So the idea of an epidemiologist is try to figure out three main things. They have three goals. The first one is to monitor the incidence of disease. And by incidence, I guess I can, if you don't know what that means, the incidence is basically the rate at which a disease is diagnosed. Right, I mean, you could have incidence more than it's being diagnosed, but if it wasn't diagnosed, you don't know that it was there, right? So incidence is the amount of times you're going to have some disease, some cancer in this case. And so they're going to monitor the incidence of a particular disease, and then they're going to use those numbers to figure out, well, what is the risk of that in the general population? So if we surveyed 100,000 people, what percentage of those people would have colon cancer, for instance? And the second goal is to try to identify groups of people whose risk is actually higher than the general population for a particular disease. So just knowing that in general, if you surveyed everyone in the United States and colon cancer was going to affect 1 out of, let's say, I don't remember what the numbers are, you probably should know these numbers, let's say 1 out of 20 people are going to get colon cancer. Are there groups of people that seem to have a higher risk than that? And then ultimately to try to figure out what factors might be responsible for that higher risk. So the ultimate goal here of the epidemiologist, again, is to study the distribution and the prevalence of this disease, and then with these 3 pieces of information to try to put together recommendations for things to avoid, for instance, if we want to try to avoid exposure to something that could increase our risk of cancer. And again, this could apply to other diseases, but we're talking about this in terms of cancer. So we've gone over, we said now that cancer is a genetic disease, right? And it's the result of mutations in our DNA. And if we think about getting mutations, you can imagine that we could get them because we're exposed to some mutagen, and we're going to talk about that a lot in this class today. But also, you could get mutations in your DNA just from the act of copying that DNA, right? So every time your cell divides, it needs to very hopefully accurately copy every base pair of DNA in the S phase of the cell cycle, and that is actually an error-prone process. And that's why we have a lot of repair pathways to try to fix the problems that happen. And so if we think, okay, is cancer just a response or a result of just the normal errors that happen during our DNA replication? Then what would cancer look like? Okay, let's do sort of a thought exercise now, right? What if cancer was only the result of our DNA being replicated every time we have to, duplicate cells in our body? Would you have data like the ones that this table I'm showing here? So let's look at this table. So this is geographic variation in cancer incidence and death rates. And what they're doing is they're showing the most extreme differences in the rate of, in this case, death, okay? And so the relative risk for the country with the highest risk versus the lowest. So let's look at the top one, skin cancer, and specifically melanoma. If you are in Australia, and in particular Queensland, Australia, for this study, you have a 155 times higher relative risk of getting melanoma than if you are in Japan. Okay, that's tremendous. That's a huge difference, right? So if melanoma was simply the response of our cells dividing and our cells making errors every time they copy their DNA, you wouldn't expect to see these differences, right? Does that make sense? Because it's not like people in Australia have cells that are dividing 155 times more than people in Japan, okay? So you can see there are this isn't just a melanoma phenomenon. Look at lip cancer, all of these cancers. So lest you think you should move to Japan because you're going to have a lower risk of melanoma, you see that Japan actually is one of the highest ones for stomach cancer. So this is the kind of epidemiological results that people look at and try to figure out, well, why? If we knew why people in Japan had higher stomach cancer, then maybe we would know a cause or something contributing to stomach cancer, right? And we could try to avoid it. So hopefully in this class, well, you're probably going to recognize that when you're studying something like cancer, ultimately you want to apply this to humans. We want to figure out why we might get cancer or how we can protect ourselves from cancer. But it's very hard to do studies on humans. And so you're left with this kind of data, right? You're left with looking at incidence of disease, things like that. So let's think again, brainstorm here, what might contribute to this difference in Australia versus Japan? What differences might there be in these two countries? Yeah. Sun exposure. So I don't know if you all know your geography very well. I had to look this stuff up. But Australia, and specifically Queensland, is this little part X'd out here, and this is a UV index. And you can see that most of Queensland is really, either extreme or very high. Well, all of it actually is either extreme or very high UV exposure. Versus Japan, which is shown up here, and a lot of it is really in this lower range of UV exposure. So they are exposed to more UV light. Can you think of any other ideas? There are other countries close to the equator, right? There are a lot of other countries close to the equator that have probably as much UV exposure as Australia. So what other things might contribute to this extremely high risk of melanoma in Australia? First about, yeah. Pigment, so maybe their skin lacks some pigment that might protect them exactly from UV. I think sort of traditionally, historically, probably a lot of people in Australia are pretty pale skin, blue eye, much more susceptible to UV light. What other things might you imagine could contribute to this? Yeah.
Very much.
Large tanning culture, exactly. So cultural things might contribute. So maybe if you're in Australia, it's the society. I don't know. I've never been to Australia, but maybe it's a big deal to try to get a tan. Sort of the flip side to that, my colleague Dr. Yoshi Azuma, has told me that in Japan, everyone's just in their offices working super hard, and so no one's going outside in the sun. So that was his explanation. It's like no one is outside. In Australia, they're all out at the beach having fun and getting tan, and in Japan, they're all working in their offices. And so that was his explanation. So anyway, the point is, there are a lot of things that contribute to this that we're not talking about just DNA replication, right, in that error. And all of these things that we're talking about really are lumped into this idea of environment and this environmental contribution. So the same thing actually, this is showing death rates actually. So this was incidence, right? This chart was incidence. And so you might think, okay, incidence is actually probably the result of the exposure to whatever it is, environmental factor is the incidence. But you might say, well, they have different health systems in these different countries, and so maybe that could impact the death rate, and that probably does contribute something, but you can get kind of similar huge variations in relative risk when you look at death rate as well. And in this case, the top disparity here is lung cancer in males. Eastern Europe, 33 times higher chance of lung cancer or dying of lung cancer, death rate of lung cancer in Eastern Europe versus the lowest one is West Africa. And we could sit here and think about why that might be the case too. Lung cancer is kind of an obvious one because cigarette smoking can contribute. Okay, so when we look at this data, it suggests that there's some component that isn't simply just either you've inherited a mutation in your DNA that's going to lead to cancer, or you have just this acquiring of mutations during DNA replication, during cell proliferation. Okay, so what we're talking about basically is environment. Now we've mentioned a couple of things that we lump into this category of environment. You want to say them out loud and let's remind us, and I'll write them on the board. What things have we said? Culture. Yeah, we have said culture. Let me break that one down a little bit. In terms of environment, let me not write that one down quite yet. Sun exposure. So like you, yeah. So UV exposure. What other kinds of things might contribute to environment?
Chemicals.
Chemicals. Yes. Can we be more global, I guess, in terms of pollution? Maybe I should not just have, maybe I should just tell you what I want. Write it down, okay. I'm being way more generic, right? So from environment, we're talking about the air we breathe, the water that we consume or that we're in, our UV exposure, our reproductive habits. So that's kind of culture-y, I guess, could be. culture kind of has to do with a lot of these things. Culture could also be food and those kinds of things. Okay, reproductive habits. These are all environment, tobacco usage. Diet. We could kind of go on and on with the list, but you get the idea. And each of these things can contribute to your chance of getting cancer. So what we've done so far is we've just looked at data from these countries and compared high to low incidence or high and low death rates. But this still doesn't, can we get one step closer to actually showing that the environment has, how would you do that experiment? To actually show that environment has a contribution. Again, this is going to be over time, you know, years and years, decades maybe even, of exposure to different things. You can't just take populations of people and say, okay, you're gonna all go sit out in the sun and you're not for 10 years and then we're gonna check. I mean, that's not ethical, right? But what we can do is we can look at different populations that have physically moved from one location to another. So that's what's shown on this graph. What they're doing here is they're plotting the data from Osaka, Japan versus people that have moved from Japan to Hawaii versus people that have lived in Hawaii and not moved from Japan, so Caucasian people that are living in Hawaii. So it's sort of three populations of people that they're comparing. And let's not think too much about the differences in time, because that doesn't really pertain to this point that I'm trying to make here. But they have different times. So let's look for the data from the same time period, this 1988 to 1992 period. Okay. And start with prostate cancer. So if you were in Japan in that time period, your cumulative rate by age 75 of prostate cancer is super low. It's 1% maybe. Okay. If you were a Hawaiian Caucasian in that same time period, your cumulative rate of prostate cancer is quite a lot higher than 1. It's maybe, what, 13%? That's a huge difference, right? Just in those two populations. So then they took this third population. What about people that had moved previously from Japan to Hawaii? And if you look at that group, that's the green group, that's kind of in the middle there. That's 7.5%. This is intriguing. This suggests that maybe something about this suggests a couple things, including that the genetics maybe isn't impacting this. Well, there's an impact beyond just genetics, right? Because you can take people from Japan, move them to Hawaii, and they appear to have a prostate cancer incidence that is moving toward getting closer to that of the people that were living there already to start with. Does that make sense? And the same trend is holding for colon. Actually, for colon cancer, it looks like it was sort of an immediate thing. Stomach cancer, well, and breast cancer, same thing for women. A low incidence of breast cancer in Japanese women, much higher in Caucasians in Hawaii, and somewhere approaching that for first generation Hawaiians that had moved from Japan. Stomach cancer is the opposite. Remember I said stomach cancer was high in Japan. So it's a high percentage of stomach cancer, very low in Hawaii. And then if you move to Hawaii from Japan, it comes down. So this is pretty strong, right? You agree? This seems to be pretty strong evidence that there's something about the environment that's affecting these cancers. Okay, so this is the kind of studies that people can do. And this is looking at populations that have been in one place and then another population that has moved to another place. If we want to take this one step further, we might look at, and some people did look at, the next generation, right? Because you would imagine that if this is a trend about environment, then these numbers might start to really get closer and closer and closer to the numbers, the incidence, death rates of the people that were actually living in that area, and that's exactly what they see. So this is another study, very similar, but they're plotting it a little bit differently. And in this case, they're comparing Japanese immigrants to California versus males in Japan. This is all males, no breast cancer, no females here. The sons of the Japanese immigrants, and then comparing everything to white people in California, which is set at 1. Okay, that's what this graph is showing us, and what you hopefully are able to see is this is graphed differently, so you have to look at it a little bit longer to see this. But let's look at stomach cancer first, because that's this big obvious blue line. Again, high chance. This is relative death rate. High relative death rate if you're living in Japan. For the immigrants to California, that decreased, and for the sons of those immigrants, it was even less. Coming down, maybe approaching that of the white people that were living in California, I guess, that they compared this to. Similar kind of trends for liver. And then in the opposite direction, because again, it's not like, Japan is going to give you, living in Japan is going to give you cancer, living in California is going to protect you. You look at prostate cancer, and in Japan, or colon cancer actually, both of those were pretty low for people in Japan. The first generation moving to California, that became higher. The second generation, it was pretty much the same as the people that had been living in California. Okay. This is classical data that you'll be exposed to again, probably, if you ever hear about environmental effects on cancer. It's very, very clear that the environment has a big impact on a lot of the cancers. Okay. But I need to caution you. Remember, I said this is a study on humans, and basically all we can do is look at the data like this. You can't set up groups of humans and have them, move populations of people just to do a study. So a lot of this is correlative, and so I want to caution you with this silly slide that basically, if we wanted to try to prevent global warming, one way of doing that, according to data, would be if we increase the number of pirates. Because back when we had lots of pirates in the world, 35,000 estimated in 1820, the global temperatures were 14 degrees Celsius. Now they're creeping up to 16 degrees Celsius, and we have way fewer pirates. Okay. Silly. But the point is, you can make correlations between a lot of things, right? And just because, okay, people moved to from Japan to Hawaii, and then they had higher lower stomach cancer, let's say, is that because of the move? What, it's kind of complicated. Okay, so I'm just going to caution you, because a lot of the human study data is correlation like that, and you have to really figure out, does that make sense? Okay, are there any comments or questions about that idea, the concept of environment? Cool. So a conclusion from all of that data, one conclusion is that most mutations that cause cancer are in fact not inherited. So they are acquired during our lifetimes in our somatic cells. Remember, we talked about the difference between germline mutation and a somatic mutation. So I guess that's a good thing, maybe, if we can somehow control our environment, or for the parts of our environment that we can control, that maybe is a good thing. So what are these environmental factors? And can we get, if we know what the environmental factors are, right, we can try to avoid them, possibly. And so we have known about environmental factors now since 1775, which is a long time ago. Before that, people really didn't know anything that was related to causing a cancer. But in 1775, this English surgeon, Percival Pott, started noticing that there was a high incidence of scrotal cancer in men that had been chimney sweeps when they were boys. So I don't know how many of you are fans of Mary Poppins or even think about chimney sweeps, but Apparently, this was a very common profession back in the 1700s, and this sign, I didn't think we even had chimney sweeps, but this sign was a block away from my house, an advertisement for chimney sweeps last summer. So apparently people still do this, but I doubt if they do the same way. So back then, they had boys as chimney sweeps because they were small enough to be able to fit into the chimney. And they'd actually go down into the chimney and clean out the soot of the chimney. And the historical reports of this are maybe a little bit varied. Some of them suggest that they weren't wearing clothes because then they wouldn't get their clothes caught on things. Doesn't really matter if they were or weren't. The point is that they were going into the chimney and getting as chimney sweeps to clean, and then later on, a lot of them were getting scrotal cancer. Okay. And so Percival Pott thought this was pretty strange, and he wanted to try to figure out why this was the case. Just to give you some context here about when this was all happening, this is when we had the American Revolution. So here we had, Paul Revere running through the streets. This is putting this all in perspective, historical perspective. So this is a long time ago, right? And so what he noticed though is he went over to Europe. This was in England. He went over to Europe and he noticed that those men that had been chimney sweeps were not getting scrotal cancer. So that was pretty strange to him, and he thought then, oh wow, well maybe what's the difference? What's the difference? What possible difference could there be? So now you all think, what kinds of things might you consider? What would be the differences between these two populations? Just throw out some ideas. Say that again? The type of wood they're burning. I think that would have been a fantastic thing. He looked at that and it didn't seem to be different. The wood that was burned in Europe versus England. That would be logical though. Maybe it has more carcinogens if you burn it. What else could possibly? Any ideas?
Differences in like protective equipment, like masks or something.
So, protective equipment like masks, no, neither of them were wearing protective equipment, and let's just assume for the sake of this discussion that they were all not wearing clothes either. Okay, that's the key point, maybe, or a hint.
The amount of time they spent doing it?
Nope, the amount of time was about the same. They would clean the same way, yeah. Say that, sorry, louder.
Sorry, how
The building material of the chimney, that's another option, but no, those were all the same, yeah?
was it the hygiene of the boys' post?
Hygiene of the boys' post, that ended up being the thing. So he figured out that the boys in Europe were bathing more often than the boys in England. So they're both being exposed to the same carcinogens, the same length of time, the same, well, no, not the same length of time, but in terms of being in the chimney, the same length of time, right? But in England, apparently they weren't bathing for a long time, like maybe a week or so. And so the soot from the chimney was on their bodies that long, and he concluded that it was that difference in time to wash off the carcinogen that was responsible for this. Okay, so contrary to Mary Poppins, chim chimney, as sweet as lucky can be. You all know that song. They weren't very lucky, right? They were getting scrotal cancer when they grew up, at least if they were in England. So now when I watch that movie, I think, oh, this is, sorry, I just can't. Percival Pott, oh, he's gonna come and Dick Van Dyke, or Lin-Manuel Miranda, I guess that's the more recent one, right? Okay, so that's again, this is the classic story of the first time anyone had figured out there was some environmental component that was related to a cancer. So Percival Pott gets a lot of credit for this idea of epidemiology. This was kind of the first epidemiological study ever, right? And it took a long time, over a century more, to start to test this idea that there was something in the soot that was causing this cancer. So Yamajiwa Back, many years later, in 1915, which is still a long time ago, but it was way past the Percival Pot era, he and his lab were taking coal tar, like the tar that's created from burning coal, and they were painting the ears of rabbits with this coal tar daily, over and over and over. After 660 days of painting this tar on the ear of the rabbit, there was a rabbit that got a tumor on the ear, right there, and there's the tumor. And this rabbit ear is preserved in a museum in University of Tokyo, so you can go there and see this. So this was then the first demonstration, an actual experiment, right? So this is just, again, epidemiology. It seems like they're bathing more. I'm thinking it's probably because they're both exposed to some chemical carcinogen, and probably they're washing it off faster, so that's it, right? So then this study by Yamagiwa, the first one where they're actually doing an experiment to show, yes, if you paint this coal tar on the ear of an animal, it gets a tumor. Okay, and then many years after that, not quite as many, only 15, but chemists started taking that coal tar. Well, what is it in the coal tar? What about the coal tar? Is it all the coal tar, or there are parts of the coal tar we can separate out? And they identified this benzoapyrene, which sometimes is just abbreviated BAP, as a carcinogen that is found in coal tar. Benzoapyrene. So the chemists dissected out the coal tar and figured out at least one carcinogen that's in coal tar. We know now that this benzoapyrene is also a component of tobacco, and it also, is also a component of grilled meats, burned parts of grilled meats, unfortunately. But let's focus on tobacco for the moment. 30%, and we also know now that 30% of all human cancers are associated with tobacco use. So many years after this original chimney sweep observation, we're left with this data. But so what is benzoapyrene? Okay, benzoapyrene is a pretty innocuous chemical. It's right here. And let me tell you at this point that most carcinogens, or many carcinogens, are electrophilic. And we're talking chemical carcinogen now, okay? Electrophilic. So what does that mean again? Refresh my memory. What does electrophilic mean? Loves what? The electrons, which is what charge? Negative charge, right? So what is our DNA? Negative charge. So most of the things that are going to be carcinogenic are ending up interacting with negative charge. Benzoapyrene is not such a chemical. I don't know if all of you have had the biochemistry class, or a lot of you have had biochemistry class, so you're looking at that and going, that doesn't look electrophilic at all. And it isn't. So what happens with benzoapyrene and with a lot of carcinogens is they start out being something called a pro-carcinogen. And that's what, benzoapyrene, which I'll abbreviate like that, is a pro-carcinogen. Okay? And what happens is benzoapyrene and a lot of other pro-carcinogens, your body takes them up and your body tries to get rid of it. But it's incredibly insoluble, okay? Insoluble. So your body has to solubilize it in order to get rid of it, get rid of this waste as waste. And so it has this series of, enzymes, which I'm gonna just abbreviate cytochrome P450, and then I'm gonna write it out in a second, that convert this pro-carcinogen into what's called the ultimate carcinogen. And in the case of benzoapyrene, that is an epoxide. is electrophilic, okay? So this epoxide is electrophilic. It's also soluble. So your body can get rid of it, okay? So in the process of your body trying to make this insoluble compound that was not carcinogenic actually in and of itself, it's not gonna attack your DNA, and it's not gonna interact with your DNA. In the process of making it soluble, your body converts it into something that is an electrophile and can interact with DNA, can cause mutations, right? So it's kind of a funny thing, sort of ironic that this enzyme that's trying to detoxify things, detoxify by solubilizing things so we can excrete them, ends up converting pro-carcinogens into ultimate carcinogens. So, for those of you who are curious about what CYP450 stands for, it's cytochrome because it has a color. So that's, what is cyto? Cyto is cellular, of course, I guess, and chrome is colored, just the root of those words. Cytochrome P450 because it absorbs F450. So that's how it gets its name. And the P is pigment. So it's basically, that's just how it gets its name. You can just refer to it as CYP450. I'm not gonna ask you what, to say cytochrome P450. But some people like to hear the whole roots of the words, just to help lock it in their brain. Okay, another key feature of CYP450 and these other cytochromes that do this work is that bacteria don't have them, and that's going to be important when we talk about the Ames test. Bacteria don't have them. So basically, if you expose bacteria to cytochrome P450, it's not going to convert it into an epoxy. Okay, let's see how far I want to get before a break. Actually, let's take the break. Let's take our break now. I can set up. Yeah, let's take a break now. And we're gonna come back and I'm gonna give you my public service announcement about smoking. So please don't right now go out and smoke. Because that will, well, yeah.
Oh, yeah.
I guess it will be before my public service announcement. Now's the time. No. I know, this is the day that I can, this is the day that I can. Sorry, the rest, yeah, got it, just a little island, I guess, got it. Didn't pay as much. At least back then, 17. You think it's still true? Okay.
If you turn it on for like 5 seconds.
Okay, I'm eager for this public service announcement. This is gonna actually take a little bit of time. So we're sort of segueing from talking about chimney sweeps being exposed to something in the soot to painting soot on ears to show it actually can grow a tumor, to a chemist figuring out, oh, what is it in there that's causing the problems with the tumor, and now we've identified benzoate pyrene, and I've told you that benzoate pyrene is part of And so now, we have known for a long time, actually since 1950, this study is pretty old, that your relative risk of lung cancer is a function of the number of cigarettes you smoke per day. This kind of makes, you have heard this your whole life probably, right? Which is interesting, that you have heard this your whole life, that cigarette smoking can lead to lung cancer, actually can lead to heart disease, all sorts of things, right? And the more you smoke, the increase of your relative risk. So there's a pretty clear, again, a correlation, but it's pretty clear correlation. If you look at the data in another way, and here what they're doing is plotting the per capita cigarette consumption in the United States. And you see in 1900, very few people smoked. It started increasing and increasing and increasing until it got to the mid 1960s when it started decreasing. Till now it's here. Per capita cigarette consumption, the legend is over on this left side. And what we see from this chart is that the male lung cancer death rate seems to be following the same trend, only displaced by maybe 30 years. So you're plotting male lung cancer death, here we go, it's coming up, it's coming up, it's coming up, and then it peaks and it's starting to come down. Female lung cancer death rate is actually lagging behind the male, and it's unfortunately still increasing. Okay, so look at this chart, and what can we make from this chart? Or what kinds of things can we, what questions do we have? One question might be, what happened in 1965 or in the mid-60s that caused this to start coming down? And that was the Surgeon General's warning that cigarette smoking was bad for us. And that happened then, and people started listening and thinking about it and decreasing the cigarette consumption. Another question is, why is female lung cancer death rate lagging behind male lung cancer death rate? Can any of you think of why that might be the case? Yeah.
Was it not socially acceptable for women to smoke as when it started compared to now?
Yeah, so two different things. One is, as you're saying, it was not as socially acceptable. In fact, it was looked at as being pretty bad if you were a female that was smoking. That was not a cool thing to be female and smoking. It was pretty cool to be male and smoking back here. The other thing that contributed to that is a lot of the people that started smoking in this time frame, they started smoking because they were fighting in wars, and they actually put cigarettes in their K-packs, their rations from the government. So not only was the government trying to keep people from smoking, the government was actually giving everyone cigarettes and saying, here. I'm not blaming the government didn't know, right? No one knew at this point, I'm assuming, I want to believe, no one knew that this was going to be such a harmful pastime, okay? To kind of illustrate that point that no one knew, I want to show you some commercials from back before the Surgeon General warning. Okay, here's one. Make it bigger. See if you can. Let me turn up the volume. I don't know how loud this is going to be. Yeah. Okay. So look at this commercial. Can you even imagine if this?
Busy doctor as he makes his daily round of calls. You'll find yourself having a mighty busy time keeping up with him. Time out for many men of medicine usually means just long enough to enjoy a cigarette. And because they know what a pleasure it is to smoke a mild, good-tasting cigarette, they're particular about the brand they choose. In a repeated national survey, doctors in all branches of medicine, doctors in all parts of the country were asked, "What cigarette do you smoke, doctor?" Once again, the brand named most was Camel. Yes, according to this repeated nationwide survey, more doctors smoke Camels than any other cigarette. Why not change to Camels for the next 30 days and see what a difference it makes in your smoking enjoyment? See how Camels agree with your throat. See how mild and good-tasting a cigarette can be.
Okay, so this is an example that.
You know, if you were to
Stop. I find this shocking, right? Because now we know there would be no commercial where doctors are recommending this particular cigarette brand, right? Okay. I'm just trying to take you back to then, because I think it helps us not get all angry with our grandparents and parents and people that smoke that's of a different generation. People didn't know. Here's another one. This is an actual cartoon that I would like to watch when I was younger. Flintstones.
They sure work hard, don't they, Barney? Yeah? I hate to see them work so hard. Yeah, me too. Let's go around back where we can't see them. Gee, we ought to do something, Fred. Okay? How's about taking a nap? I got a better idea. Let's take a Winston break. That's it! Winston is the one filter cigarette that delivers flavor 20 times a pack. Winston's got that filter blend. Yeah, Fred. Filter blend makes the big taste difference, and only Winston has it up front where it counts. Here, ahead is a pure white filter. Winston packs rich tobaccos specially selected and specially processed for good flavor and filter smoke. Yeah, Barney, Winston tastes good, like cigarette should. The Flintstones have been brought to you by Winston, America's best-selling, best-tasting filter
And now
cigarette.
actually, Wilma's also smoking.
Like a cigarette should.
Okay. I don't remember those commercials. I watched the Flintstones, but I don't remember those commercials. But anyway, I suppose I saw that and it's just like, oh yeah, people smoke. Barney and Fred are smoking. Okay. So these were earlier in the 50s, I think. In the 60s, the later 60s, it became kind of a women's liberation thing for women to start smoking. And so now women are going to be smoking, and this is how they advertised to women.
Back in the old days, men were the masters, women were the slaves. Women had the duties, men had all the rights. Then at last, women won their rights, and one by one, they won them all. You come a long way, baby, to get where you got to today. You got your own cigarette now, baby. You come a long, long way.
Introducing new Virginia Slims, the slim cigarette for women only, tailored for the feminine hand. With flavor women like, mellow, mild Virginia flavor. New Virginia Slims in the slim purse pack.
You got your own cigarette now, baby. You come a long, long way.
Marketing, right? You've got your own cigarette. Whoa. And then, yeah. That's because it's a YouTube video now, so now we have to put that on. That was not on back when this was showing originally. Anyway, I play this every year at home the night before I teach this class, and my kids know that song. My daughter came into the room and I was like, you got your own cigarette now, baby. It's like, oh my stars, I totally warped my daughter at least. She knows the song. Hopefully that's not going to, yes, close, all tabs. Okay, so the point is, cigarette was a big deal, and you might know all this, but cigarette marketing was huge. First men smoked, then women smoked. This is all correlation, but it seems like, about a 30-year lag, women weren't really smoking much here. Then women started smoking in the 60s, now women are getting lung cancer. These are the links to those videos if you want to just watch on your own, because they're kind of hilarious. It's hard to imagine, though. I even find it hard to believe a doctor, promoting, advertising a cigarette. They didn't know that they were going to be harmful. So now the unfortunate thing is, as I was saying before, this is the actual data. This is stats for women, for death rates, coming down for liver and stomach and uterine cancer, colorectal, breast, it's all coming down. But lung cancer is not. Lung cancer, maybe if you want to try to be optimistic, is slightly coming down, but I've read things that say it's still really not coming down. It's kind of staying there for women. And so, you've come a long way, baby, but we need to all be thinking about not smoking. So if you don't already smoke, then don't start, because I have seen in my friends and roommates that it's super, super hard to stop. And so that would be my public service announcement. If you can get your friends to stop smoking, it's gonna be better for their health to not be smoking. And the good news about stopping smoking, here you go, here's the little bit of glimmer of hope. This was an the American Cancer Society study, looking at people that had smoked either 1 to 19 cigarettes a day or 20 plus cigarettes a day, and comparing their mortality ratio of lung cancer to those that had to current smokers. And actually, the mortality ratio of 1 would be never smokers, somewhere on here. And what we see is that the longer you can stop smoking, your lungs actually can recover. And so you can get back to having a lower chance of dying of lung cancer, back to almost as if you hadn't smoked before. Now, I hesitate to tell you this because you all are young and you're gonna say, okay, then I can smoke now for 20 years and I'll stop, and then, it's gonna be a matter of whether that benzoylpyrene and the stuff in the cigarettes now just happens to hit the right tumor suppressors and oncogenes in your youth, and, so again, this is the public service announcement. Being a cancer researcher, lung cancer and smoking cigarettes is the one. Like, if we could stop everyone from smoking, we would make a huge impact on cancer, not actually just lung cancer. So that's the thing. If I could help some of you encourage your parents or friends to not smoke, I would be doing something. Good. Okay, so that was that. Any questions or comments about cigarette smoking? Okay. And I'm not going to talk about vaping because I don't know enough about it, but it's one of these things that's new, kind of like cigarettes were new and we didn't really know the health impacts. I think with vaping, it's going to take some time for us to know. They're starting to get some data that maybe it's not completely healthy, but I'm not going to come out on either side of that yet because I haven't read about it enough myself. But you all are scientists, you can do the reading, you can figure out whether you want to start that or not. Okay. So we're going to talk now about, real briefly, another pro-carcinogen. So I talked about the one, benzoylpyrene. This is aflatoxin. Okay, so this is a pro-carcinogen. Remember, which means it isn't a carcinogen in and of itself. It needs to be converted into one in your body by like cytochrome P450s. It's produced by mold. A specific mold, and I'm not going to write down the name of the mold because you don't need to know the name of the mold, but it's produced by this mold, Aspergillus flavus. So it's a specific type of mold. And it's often found on peanuts and rice that are stored improperly. And we suspect, again, you can't do the studies to actually demonstrate this because you can't just expose people to aflatoxin. But epidemiological studies suggest that maybe aflatoxin is associated with liver cancer, okay? What you can do is you can give aflatoxin to a rodent, and they have done rat studies, and in rats, it results in liver cancer. Okay, so we could give this to a rat, it'll get liver cancer. We know that it's found in this mold, and that mold is found on rice and peanuts, and there often are the same contaminants of rice and peanuts in regions of Asia and Africa, and those same places also have a higher incidence of liver cancer. Okay, so maybe we can put this data together and suggest that in humans, aflatoxin also can contribute to liver cancer. But this is all the data we have, right? We know it does in rats. It's present there on the ricin peanuts that people consume in these areas, and they have a higher incidence of liver cancer, so maybe it's all correlated. There's one confounding thing, though, in these same regions of Asia and Africa, there's also a higher incidence of a virus, hepatitis virus. So, also high hepatitis virus, which we're going to talk about when we talk about viruses that can contribute to cancer, and we think that this one can also contribute to liver cancer. So this part of the hepatitis is liver. And so it's hard to know exactly how much aflatoxin contributes to liver cancer, but that's the data. So it's thought that it probably does somehow correlate with or contribute to cancer. Again, as a pro-carcinogen, not as a ultimate carcinogen. Is that? Okay. So I said that a lot of these pro-carcinogens become something that's called the ultimate carcinogen, that is electrophilic. And so I wanted to show you an example of what that might look like in terms of with that electrophile interacting with DNA and what would be the consequence. So this isn't the epoxide from benzoate pyrene or the ultimate carcinogen from aflatoxin, but this is actually a common lab mutagen that we use in research studies, but it is also electrophilic. And what it does, what EMS does, is it adds, here's guanine here, and what EMS does is it can add an ethyl group, shown up here, to the guanine. And so now it results in O6-ethyl guanine. The problem with O6-ethyl guanine is that it doesn't look like guanine anymore, and in fact it doesn't base pair, it can't base pair with cytosine anymore like it was supposed to. And so the next round of DNA replication, when this DNA is being copied, and the O6-ethyl guanine now is read, and inserted across from it is a thymine, because that's what they pair with that. Okay? So ultimately, after a round of replication, you end up going from a guanine cytosine base pair to this O6-ethyl guanine thymine. So it's a GC to AT transition. So you can see why this would cause a mutation, right? It's causing what was a GC base pair to now become an AT base pair after replication. So this is kind of the idea with these electrophiles. They somehow attack, or they interact with, they modify the DNA, and then that changes the properties of that DNA so that now it resembles another base. Okay. So what we're talking about here now, I've sort of transitioned into talking about a mutagen. And many carcinogens are mutagens, meaning many carcinogens bind to DNA, somehow interact with it, and cause DNA mutations. And it turns out that there are chemical mutagens, and that's what we've been talking about now, chemical mutagens. There are also physical mutagens, or physical carcinogens that can act as mutagens. Maybe I should stop right here and kind of define what I'm talking about when I talk about a carcinogen versus a mutagen, okay? A carcinogen, in order to show that something is a carcinogen, you have to show that it can actually cause a tumor. So, I've shown you one study like that. Does anyone want to remind us all what that one study was that I showed, where someone demonstrated that a carcinogen was a carcinogen?
Coal tar on the rabbit ear.
Coal tar on the rabbit ear, right? That's a study to show that it is a carcinogen. You painted it on an animal, it caused a tumor, it is a carcinogen, right? That's the definition. A mutagen, on the other hand, what it needs to be a mutagen is just mutagenize, make a mutation in the DNA. And I'm going to show you an assay, the Ames test, that assays for being a mutagen. Those aren't necessarily the same, okay? Very, very likely though, if something acts as a mutagen, it could probably act as a carcinogen because it's mutating the DNA. but the opposite isn't always the case. Okay, but now we're kind of moving slightly, coming out in a little bit of a detour and talking about physical carcinogens. So these are not chemicals, these are physical. This is like X-rays and radon gas. Okay, so basically in the late 1800s, we have the invention of X-ray tubes. A little bit later, physicians and researchers working with those X-rays experienced increased rates of cancer. You do not need to remember the dates, but just kind of think about the order. This is historically the perspective of how this was figured out, that we now think that X-rays can be carcinogenic. In 1924, they found that there was high concentration of a radon gas, so radioactive gas, that miners were being exposed to in Germany and Czechoslovakia, and these miners had lung cancer, and historically they recognized, looking back, oh yeah, these miners have had lung cancer for a long, long time. Centuries of miners getting lung cancer, and then in the 1920s they figured out that the gas actually had radioactivity, the radon gas that they had been breathing. But this is still sort of correlative. And then in the 1920s, 1927, Hermann Mueller found that he could actually induce a mutation using a model organism, the fruit fly, by exposing them to X-rays. So this was the first experiment now, where we're taking the X-ray and we're putting it on a fly, and now we can see that the DNA becomes damaged. There's a mutation. They did similar kinds of studies a little bit later using chemicals on flies. Okay. So this is all showing, again, to be mutagenic. This study right here isn't showing it to be carcinogenic, right? They would have had to, in this case, show that the fly grew a tumor, and then maybe they could have said it was carcinogenic in fruit flies. But this is the sort of the background historical perspective on radiation as a carcinogen. Okay, so now we know that, getting back kind of to the environment, what all is contributing to our possible risk of getting cancer. This is some of the latest data, I guess, in terms of percentages. And if you look here, the main one is tobacco use. It's estimated that about over 30% of the cancers have a contribution of tobacco to that cancer. The relative contribution to cancer incidence for tobacco use is a little over 30%. You can look at the slide yourself and see different things. Diet is down here, alcohol here, physical inactivity is here, viruses and bacteria are right here. This next one I think is an interesting one because I think it's really, people are studying this now to try to figure out what is the connection exactly. What is it about obesity, or is it maybe sugar? Which is super unfortunate for me because I love sugar, and sugar is, if I had to, I don't smoke, so fine. I can stand on my soapbox and say, don't smoke, don't start to smoke. But there's enough evidence out there that sugar isn't good for us, and I can't stop eating sugar. I mean, it's tough, right? If there's something that you really like. But I think it's not good for me, and I think it's probably not good for any of us. And so I think that that's probably going to be a lot of this. I don't know that it's necessarily being overweight or obese, but maybe the sugar intake. This is my own sort of thinking here. Anyway, so what are we going to do? We're going to try to just sort of do everything in moderation, I guess. I'm not sure. Here's another table about known and suspected causes of human cancer from the textbook now. Occupational exposure isn't 1 to 2%. Tobacco related is about 1/3 again. They're saying they split it out into diet low in vegetables, high in salts, or diet high in fat is contributing to these. Here they're saying diet. That's why I wanted to show you. This is saying obesity, overweight on this one, but this is splitting it out more into diet contributing, tobacco plus alcohol. So those are things that maybe we can control to some extent. Maybe we can try to eat more vegetables. We can try to eat less fat, less sugar probably. Some of these things are maybe easier to control. We're just not gonna be chimney sweeps, none of us. None of us are gonna be chimney sweeps. These are the different carcinogens, or the exposures that are really strongly implicated in the cause of certain cancers. So we've got the scrotal cancer and the chimney sweeps. We have radium and osteobone cancer and exposure to radium. They used to have shoe fitting things where you put your foot in and it's like a x-ray to see the size of your foot. A long time ago, like my grandparents' generation, I think they realized that wasn't a good thing. Asbestos, mesothelioma, you can look on here. And we're going to talk about this one, diethylstilbestrol, here in just a little bit. Okay. But before we do that, we're gonna go over the Ames test. Oh, well, before we go over the Ames test, so two befores. It's estimated now that maybe 60 to 90%, so it depends on the study, but maybe 60 to 90% of cancers are due to environment. So that's seems kind of like a lot, right? A lot of percentage of things that maybe we can control some. Some of it we can't control still, even though it's environment, we can't control it. We're probably not going to control where we live, for instance. Okay, so how do we figure out if there's so much due to environment and some of this are mutagens? That makes sense. If there's a mutagen that we're consuming and that's causing cancer, we want to know about it. So how are we going to figure out what those are? So there's this test that was developed called the Ames test, and it's a quick and easy way to detect a mutagen, okay? So the Ames test is detecting mutagens, again, not carcinogens necessarily, but mutagens. And the way it works is you take a test compound, let's say I wanted to see if the seed of an apple was mutagenic. I could grind up the seed of the apple and I could add it to some salmonella bacteria. Let's ignore this part for now, okay? Add it to some salmonella bacteria that were unable to grow without added histidine in the culture medium. So what's happened with these bacteria is they have a point mutation that makes them unable to produce histidine. Okay, so you need to provide that in their food or they cannot grow. Does that make sense? I'm gonna write that on the board somehow. So these bacteria have a point mutation that they cannot grow without histidine. So you have to plate them, you have to put them on food that has histidine in it. And then the idea is you mix those bacteria with your compound you're testing, and then you see how many, you let them grow, and you see how many, you can just count the number of bacterial colonies. And each of those bacterial colonies represents a bacteria that had a mutation that now converted that mutant gene for histidine back into being able to make histidine. So it's a revertant. So this, I'll write it on the board. Okay. So they have a single base pair mutation. I'm going to abbreviate base pair that way. That makes them unable to produce histidine. Okay. If you add a mutagen to them, then some of them are going to have this revert, right? If you add a mutagen, some percentage are going to have a reversion, and now that's going to be able to make histidine. Okay, and then you can just count the colonies. So it's a really, really simple assay. So I've explained this part of the assay. Why are they adding the rat liver extract? Does anyone want to guess? Or remember, or have learned about this before? Yeah.
We're trying to scan out for like pro-carcinogen.
Yeah, exactly. So remember that idea about a pro-carcinogen not being able to be, isn't an ultimate carcinogen, it has to be converted to one. It's kind of the same idea in terms of mutagens, right? I don't think there's a term pro-mutagen though, but it's the idea, it's the same idea. These bacteria, remember, they didn't have any CYP450. They don't have cytochrome P450. And so let's just say that this test compound was the benzoate-pyrene. You put that on the bacteria, they're actually not going to, it's not a mutagen. It's not an electrophile, it doesn't touch the DNA, okay? So what they do for this assay is they add a rat liver where there is cytochrome P450, right? So they're providing the cytochrome P450 in this rat liver, and they mix that up with their test compound like benzoate or the apple seed, whatever. And then they add that to the bacteria and look to see if it's mutagenic. All right, this test is very easy, very simple, cheap, quick. You can screen through lots of compounds and lots of test things really, really easily. Does this assay make sense, everyone? Okay, good. When you do this assay, but remember, this assay was looking for mutagens, not carcinogens, but mutagens. Looking for mutagens. If you think logically, probably if it's going to cause mutations, very likely it will also cause cancer, but it isn't testing for that, right? We're not looking to see if a tumor grows on anything. But when they do what's shown here on this slide is plotting the Ames test results, and the way this is shown is the micrograms of whatever this test compound was required to induce 100 Salmonella revertans. So basically, the smaller number, the more mutagenic it is, right? If it just took 10 to the minus 3 micrograms to make 100 Salmonella revertans, it's down here. Here it took 100. They've plotted this on the x-axis and on the y-axis. They're plotting these same compounds now for an actual carcinogen test, where they're putting them in a rodent model and they're looking to see how much of this compound it took to give half of the animals a tumor, an actual tumor. So basically, you're plotting carcinogenic potential, mutagenic potential here, and you see a pretty nice correlation, right? And so I'll point out the one that's the highest, the worst here, and which is one of the reasons why I talked about it in class, is this aflatoxin B1. This is a really potent mutagen. It doesn't take very much to make mutations in these bacteria, presumably also in us. It also doesn't take very many milligrams per kilogram of rodent to make a tumor in an animal. So that's that one. Benzoate-pyrene's kind of in the middle, still not, none of these are good, but there's a nice correlation between mutagenic potential and carcinogenic potential, okay? So when this test it's really easy and cheap, as I said, to do, and so scientists were doing this test on everything they could get their hands on to try to figure out, well, what should we be eating then, right? What is it, a mutagen or not a mutagen? And here's a list of things that are identified. I don't like that this is from the book, and now this is the first time I think I'm noticing that this is calling this a carcinogen, but if it's the Ames test, it really should be just a mutagen. But anyway, these are things that were identified using this Ames test as should be mutagenic, but they've also registered, I guess, okay, if I read the fine print, they're also registered in laboratory animals as carcinogen, because that's the only way we're going to test if they're carcinogenic. So look at this stuff. These are things that caffeic acid in coffee. I drink coffee every day. Cocoa, I love chocolate. Rhubarb, we eat these things, and this whole list of things are things that we're consuming and we're supposed to consume. So the bottom line with this, I think, is that basically there are mutagens all over the place, and we're going to have to you have to figure out how much of whatever it is you're consuming, because we have to eat something in order to survive. I guess I'll leave you on that happy note.